All right, thanks, Erica. My name is Brittany Hartwell, and I'm very excited to be here with you all today to share some about this image that I collected as a postdoc in Daryl Urban's lab, along with Jason Chang, another postdoc in the Urban lab who's not able to be here today. In the Urban lab, we work on developing vaccines and immunotherapies that can train the immune system to fight disease, ranging from cancer to COVID. This particular image shows a vaccine that we engineered to be taken up across mucosal tissues in the nose in order to activate a stronger immune response to protect against infectious diseases. So why might we want to engineer vaccines that can target mucosal tissues like this? Well, it's because intuitively we know that most pathogens are transmitted by mucosal routes. Take SARS-CoV-2, for instance, the virus that causes COVID. It's transmitted via the upper and lower respiratory tract. HIV is transmitted via the genitourinary tract. So it makes sense that an ideal vaccine would establish immune protection at this mucosal tissue as a front line of defense. But now consider most of the vaccines that we receive when we go to the clinic, they're injected. And this sort of traditional injected vaccine, while very good at establishing immune protection to protect us from severe disease and severe infection, they actually aren't really designed to establish immunity in the mucosa. They establish what we could refer to as a backup defense in the blood. So what we wanted to do was engineer a vaccine that could establish this frontline defense. And one way to do this is to directly administer vaccines to mucosal tissues. All right, so if this strategy of mucosal immunization can establish this enhanced immune protection, why can't we receive, say, an intranasal vaccine every time we go to the clinic? Ultimately, it comes down to a drug delivery problem. Our mucosa is designed to keep things out. So if we were delivering a vaccine by this route, ultimately we'd want to get that vaccine through these mucosal barriers into the immune cells associated with that mucosa that underlie all those barriers. In the nose, this is called the nasal associated lymphatic tissue, or the NALT. And these immune cells are really where antigen-specific immune responses against invading pathogens are orchestrated. But as you can see here, in order to get vaccine components to that NALT, first they have to overcome the mucosal barrier. Typically, antigen components are broken down or cleared away in the mucus. If they get past that barrier, then they're faced with the epithelium and tight junctions between these cells. Ultimately, this is designed to keep stuff out. But we know that a few things can get through. And one of those things is called albumin. It's a naturally occurring protein that's found in mucosal secretions. And albumin is able to get up across these barriers by binding to a receptor that then shuttles it up across the epithelium and kicks it out on the bottom underneath these other cellular layers near the NALT. So we thought, what if we engineer a vaccine that can bind onto albumin, using it as a chaperone to effectively hitchhike using this receptor to get up across these barriers. And by doing that, we could concentrate our vaccine, bypass the barriers, and concentrate our vaccine where those important immune cells are located. And when our vaccine can interface with those immune cells in the presence of antigen from our vaccine, those immune cells undergo multiple rounds of crosstalk, leading to activation of important immune cell players, which can then actually pass back out, migrate into the mucosal tissues where they're established as sentinels or guards to protect against invaders like viruses. And when they encounter those viruses in the mucosa, they secrete protective immune proteins called antibodies that can then bind onto that virus, preventing it from infecting those cells in the first place. And that is actually what we're looking at in this image that's shown in the gallery. We took this image because we wanted to see how our vaccine was behaving after it was delivered intranasally. This is showing a cross-section from a mouse's nose. The mouse was immunized with our vaccine, and 24 hours later, we took this image. The vaccine is showing up here in yellow and pink. All the cells in the mucosal tissue are shown in blue, and the airway or the negative space is shown in black. And the first thing to note here is that this image, again, was taken 24 hours after the vaccine was administered. And we can see a lot of vaccine lining the airway and being taken up into that mucosal epithelium, which indicates that first, our vaccine bypassed that initial barrier of mucosa. It's mucus. It's evaded clearance and degradation by the mucus. And here we see it actually being taken up, binding and being taken up into those mucosal epithelial cells. And if we look closer, we can see it being taken even deeper in those little vesicles and bubbles, the yellow and pink, into those underlying submucosal cell layers as well. And the reason this image is so important is because it shows that our vaccine is able to overcome these barriers for delivery, which ultimately allows it to activate better immune protection at the front line. This is another way of visualizing this. In this case, the cross section is shown with cells in blue and the vaccine is shown in red. And we can see zoomed in on those epithelial layers, the vaccine layering the epithelium being taken up in vesicles underneath. In contrast, if we compare this to a vaccine that lacks this unique engineered property of albumin binding, we don't see any vaccine retained in the airway, on the epithelium, or in the underlying cell layers. But engineering a vaccine that can get across these barriers leads to it trafficking into the nasal-associated lymphatic tissue, where it interfaces with immune cells, can activate those immune cells to then go back out, into the mucosal tissues in order to establish mucosal immunity in, for example, the nose, the lungs, or the genitourinary tract. And our research with this vaccine platform so far has shown some really promising results, showing that we can establish functional immune protection against HIV, for example, in the genitourinary tract, or against SARS-CoV-2. Against SARS-CoV-2, we've shown that we can activate antibodies in the nose and in the lungs that can neutralize virus before it's able to infect cells. And so thank you so much for your time. And I'd also like to thank my postdoc advisor, Daryl Urban, and also Jason Chang, the other postdoc who helped collect this work, and a few of the cores here at the Koch Institute who helped in the collection of this image, especially the histology facility in Kathy Cormier. Thanks so much.